Dr. C.S. Kulkarni, MD, and I am going to teach medical biochemistry. Before I start the topic, please like, share, and subscribe the channel. In the last session, we dealt with factors influencing enzyme activity. Today, we will deal with the same. The next factor is key enzymes. Body utilizes allosteric enzymes for regulating metabolic pathways. Such a regulatory enzyme in a particular pathway is called key enzyme or rate limiting enzyme. The flow of the whole pathway is considered as if there is bottleneck at the level of the enzyme. The allosteric inhibitor is most effective when the substrate concentration is low. This is metabolically very significant. When increased substrate molecules are available, there is decrease necessary for stringent regulation. Two best examples are given in detail in below. First example is succinyl CoA plus glycine in the presence of enzyme ALA synthase gives delta amino levinic acid. This is the first product step in heme synthesis. The end product heme will allosterically inhibit the ALA synthase. This enzyme is the key enzyme of heme synthesis. Next example is carbamoyl phosphate plus aspartate in the presence of aspartate transcarbamylase give carbamoyl aspartate and inorganic phosphate. This is the first step in the pathway which finally produces cytidine triphosphate. CTP, the end product, will allosterically inhibit the aspartate transcarbamylase. There is no such resemblance between aspartate and CTP. Here are a few examples of enzymes and their allosteric inhibitors and activators. For ALA synthetase, heme is an inhibitor. For aspartate transcarbamylase, heme is an inhibitor and ATP is an activator. For HMG CoA reductase, cholesterol is an inhibitor. For phosphofructokinase, ATP and citrate are inhibitor, while AMP and fructose 2,6 bisphosphate is an activator. Pyruvate carboxylase ATP is an inhibitor, whereas acetyl CoA is an activator. For acetyl CoA carboxylase, acyl CoA is an inhibitor, while citrate is an activator. For citrate synthase, ATP is an inhibitor. For carbamoyl phosphate synthesis, NAG is an inhibitor. For carbamoyl phosphate synthetase 2, UTP is an inhibitor. Coming to the next factor, feedback inhibition. The term feedback inhibition or end product inhibition means that the activity of the enzyme is inhibited by the final product of biosynthetic pathway. For example, A is converted to B in the presence of enzyme E1. B is converted to C in the presence of enzyme E2, and then C is converted to D in the presence of enzyme E3. In this pathway, if D inhibits E1, it is called feedback inhibition. For example, AMP inhibits the first step in urine synthesis. Usually such end product is affected allosterically. Here we can see that IMP finally produces EMP. Then EMP allosterically inhibits an enzyme which forms adenylosuccinate. Induction. Next factor. Induction is effected through the process of de-repression. The inducer will relieve the repression on the operator side and will remove the block on the biosynthesis of the enzyme molecules. Classical example is the induction of lactose utilizing enzymes in the bacteria when the media contains lactose in the absence of glucose. There will be a minimum level of the enzyme inside the cell, but in the presence of the inducer, the enzyme level will go up 1,000 or a million times within hours. By this mechanism, nutrients are utilized most efficiently, and while the enzyme synthesis is kept to the optimum. Here are certain examples and their inducers. Tryptophan, pyrrolase, and transaminases. Their inducer is glucocorticoids. For glucokinase, insulin is an inducer. For ALA synthase, barbiturates is an inducer. Coming to the next factor, repression. Even though both inhibition and repression reduce the enzyme velocity, the mechanisms are different. In case of the inhibition, the inhibitor acts on the enzyme directly. The inhibitory activity is noticed as soon as the inhibitor is added, and the number of enzyme molecules is not changed by the inhibitor. On the contrary, repressor acts at the gene level. The effect is noticeable only after a lag period of hours or days. The number of enzyme molecules is reduced in the presence of repressor molecule. In this diagram, we can see that regulator gene produces aporepressor. This aporepressor plus co-repressor together form holorepressor. This holorepressor induces operator gene. This operator gene then allosterically inhibits structural genes and finally inhibits ALA synthase, which is responsible for heme synthesis. The key enzyme of heme synthesis, ALA synthase, is regulated by heme by the means of repression. The structural genes is transcribed and later translated to produce the enzyme molecules. When the heme is produced in plenty, heme acts as a co-repressor, and in combination with the aporepressor, heme will shut off the operator site. Now further production of ALA synthase is stopped. Coming to the next factor, covariant modification. The activity of enzymes may be increased or decreased by the covariant modification. It means either addition of a group to an enzyme protein by a covariant bond or removal of a group by cleaving a covariant bond. Zymogen activation by partial proteolysis is an example of covariant activation. This is a reversible reaction. Commonest type of covariant modification is the reversible protein phosphorylation. The phosphate produced group may be attached to serine, threonine, or tyrosine residues. When hormone binds to the membrane-bound receptor, hormone receptor complex is formed. The enzyme adenylate cyclase is activated. This activation is mediated through a G protein. Here in this diagram, we can see that substrate is converted to product in the presence of active enzyme. This active enzyme can be covariantly modified to an inactive enzyme EB. The active adenylate cyclase converts ATP to cyclic AMP, which acts as a second messenger. It activates both protein kinase, serine or threonine kinase by binding to the regulatory subunits of the enzyme. The active catalytic subunit will phosphorylate the enzyme. Here in this diagram, we can see that protein in the presence of enzyme kinase gets converted to protein phosphoserine. This protein phosphorylation can undergo reversible reaction in the presence of phosphatase enzyme and produce protein. The active catalytic subunit will phosphorylate the enzyme. In some cases, the receptor itself acts as tyrosine kinase activity, which is switched on when hormone binds to receptor. Example: insulin. It gives a partial list examples of such activation by phosphorylation and dephosphorylation. Here is an example of some certain enzymes who are inactivated by phosphorylation: acetyl-CoA carboxylase, glycogen synthase, pyruvate dehydrogenase, HMG-CoA reductase, pyruvate kinase, and phosphofructokinase are enzymes that get inactivated by phosphorylation. On the contrary, here are certain enzymes that get activated by phosphorylated enzyme: glycogen phosphorylase, citrate lyase, phosphorylase B kinase, HMG-CoA reductase, fructose 2,6-bisphosphatase get activated by phosphorylation. ADP ribosylation is another covalent modification where ADP ribose from NAD+ is added to enzyme or protein. For example, ADP ribosylation of enzyme subunit of G protein leads to inhibition of GTPase activity. Hence, G protein remains active. Cholera toxin and pertussis toxin act through ADP ribosylation. Coming to the next factor, stabilization. Enzyme molecules usually undergo wear and tear and finally get degraded. Such degradation is prevented that can lead to increased overall enzyme activity. This is called as stabilization of enzyme. Degradation of tryptophan pyrrolase is retarded by tryptophan. Phosphofructokinase is stabilized by growth hormone. Enzymes having SH groups like papain, urease, or succinate dehydrogenase are stabilized by glutathione. Coming to next factor, compartmentalization. The activity of the enzymes catalyzing the different steps in a metabolic pathway may be regulated by compartmentalization of enzymes. Certain enzymes of the pathway may be located in mitochondria, whereas certain other enzymes of the same pathway are cytoplasmic. Coming to the specificity of enzymes. In that first, absolute specificity. Some enzymes are absolutely specific. For example, hydrolysis of urea to ammonia and carbon dioxide is catalyzed by urease. Urea is the only substrate for urease. Thiourea, though structurally not similar to urea, will not act as a substrate for urease. Similarly, glucose oxidase will oxidize only beta D glucose and no other isomeric form. Next is bond specificity. Most of the proteolytic enzymes are showing group that is bond specificity. For example, trypsin can hydrolyze peptide bonds formed by carboxyl groups of arginine or lysine reduced in any protein. Third is group specificity. Only enzyme can react in the same reaction on a group of structurally similar compounds. Example, hexokinase can catalyze phosphorylation of glucose, galactose, and mannose. Next is stereospecificity. Human enzymes are specific for L amino acids and D carbohydrates. Fumarase will hydrate fumaric acid, that is transformed to malic acid, but the corresponding cis form malic acid will not be acted upon. In this diagram, we can see that fumaric acid, that is a transform in the presence of fumarase enzyme, is converted to malic acid, but malic acid, that is the cis form, is not converted. Here ends our session. Kindly like, share and subscribe the channel. Thank you.